Welcome to the 14th Target ALS Annual Meeting. Some of you have been to this meeting before, and for many of you, this is your first experience of this unique gathering. The presence of both these groups is a reflection of the dynamic nature of Target ALS ecosystem, and it is a joy to see friends, old and new. We are proud that all constituencies involved in ALS research worldwide are represented at this meeting. These include people with lived experience, scientists and clinicians from academia, from pharma biotech, venture capital firms, and nonprofits working on ALS and related neurodegenerative diseases. We are especially proud of two strong trends that have been reinforced over the last year. The first is an expansion of the domains of expertise that have become part of our ecosystem. As scientific advances have happened through cutting-edge research from academia and pharma biotech, we have seen incredible progress on ALS, with the first cracks in glass ceiling for ALS therapeutics. But we all know we are not yet where we all want to be. And so we need to continue to explore new horizons and try new approaches. And this will require that we include experts from other scientific disciplines and disease areas. And at Target ALS, we continue to engage with experts from other scientific domains. And you'll hear from many of them over the course of the next few days. The second strong trend is an active engagement of historically underrepresented communities worldwide. Our ALS Global Research Initiative was launched with the belief that all communities around the world should benefit from progress made on ALS, and that inclusion of data sets from diverse groups will help lead to new discoveries of our mechanisms and targets for the disease. And their participation makes our mission a more representative, a more human global effort. Now, in the few minutes I have left, I want to talk to you about a few changes at Target ALS. Hopefully, you've had a chance to look at our website and the resources we have to offer, so I'm going to focus my remarks on our board of directors, a group that is central to the overall formulation and execution of strategy for the foundation, while they leave evaluation of grants and all funding decisions to the Independent Review Committee, or IRC. So for the entirety of our existence, we have had the benefit of two board members who brought to the table invaluable experience as former directors of NINDS. And after making key contributions over the years, sadly, each have decided to step away. Story Landis wanted me to share that her decision should be taken as a compliment to Target ALS because of how we have grown over the years and that our programs are in good hands. We respect her decision and thank her for her leadership. Zach Hall has been a guiding force for Target ALS since we launched in 2013. He was scientific advisor to Dan and is one of the intellectual founders of the foundation. His wisdom allowed an original idea to transform into this globally impactful organization that we have become today. During this period, in a cruel twist of fate, Zach's wife, Julie, an incredibly warm person and a brilliant musician, was diagnosed with sporadic ALS and sadly passed away in 2022. A very personal reminder to all of us at Target ALS that this disease can affect anyone, anywhere, at any time. And at that time, it was inspiring to see Zach renew his commitment to continue to guide Target ALS. More recently, he has shared that he would like to step away and enjoy retirement. On a personal and professional front, it has been a privilege of a lifetime to get to spend time with Zach and to work with him. I'm going to immensely miss his wise counsel, his warmth, his incisive intellect. Thankfully, he has agreed to continue to answer my calls as long as they're infrequent. Sometimes words are not enough to express our gratitude, so on behalf of Target ALS, I'm simply going to say, thank you, Zach. And if I can request Zach to please, if you could stand up and wave so that everyone knows where they can find you. And I hope all of you will join me in giving Zach a round of applause, so thank him. Thank you, everyone. And it's my pleasure to invite our founder, Dan and Alisa Doctoroff, to say a few words. Thank you.
ALS has taken a lot from me in the past year. I've lost my voice. I've lost the ability to eat most things. My hands and arms have weakened, and my neck muscles have deteriorated. But I will adapt. I use eye gaze technology to communicate. I use a feeding tube to eat. I can still control my wheelchair. I'm happy, I'm resilient, optimistic, hopeful. My family gives me joy. I look around this room and I see so many people dedicating their lives to solving ALS. We have a purpose with Target ALS, and every single one of you is helping to build a world where everyone lives to reality. Across continents, disciplines, and communities, Target ALS momentum is taking hold. And it's accelerating progress in ways we have never seen before, but none of this is happening by chance. It is the result of a deliberate, coordinated strategy made possible by all of you. Your partnership, your belief in this mission, and your willingness to commit to ALS research is what drives this work forward. Together, we have built what is now the largest and most impactful organization dedicated to ALS research in the world. And we continue to refine our scientific strategy, pushing ourselves each year to be bolder, more strategic, and more collaborative. And we are just getting started. There is still much to do. And I am more committed than ever to solving this disease because we are closer than we have ever been to changing what an ALS diagnosis means. With urgency, partnership, and unwavering determination, I believe we are shaping a future where everyone with ALS lives. Thank you for being here and for being part of this work.
Good morning. I am so happy to be in this room again with its buzzy vitality, ideas flying around, progress being shared. Because I need some hope. I need some inspiration. Besides from Dan, who is very inspiring. Over the last several months, we've lost 3 good friends to ALS. First, Eric Dane. He died in January at age 53, leaving behind 2 young children and a hugely successful acting career. He was diagnosed with ALS in the spring of 2025 after having symptoms for over a year. Known as Dr. McSteamy for his longtime role on Grey's Anatomy and more recently as Cal Jacobs on the series Euphoria, Eric used his public platform to join the fight for ALS awareness and ALS cure, even appearing in a TV show, Brilliant Minds, as an ALS patient. Eric became a friend of Dan's and a board member of Target ALS. Second, Leah Stavenhagen. She died in February at age 35, leaving behind her husband Hugo, mother, father Jeff, who may be in the room, but I know will be here, and 2 sisters. Leah was diagnosed at 26 and lived with ALS for 9 years. Leah, who I met at a previous annual meeting, she was a force in the ALS world and in general. Creating her ALS story, an organization to bring young women with ALS together for strength, for support, for community. There are now 150 members worldwide. she continued to travel throughout her illness, which was a passion of hers. Before her diagnosis, she loved to dance and swim. She wrote a book called I Would Love to Dance Again. Third, Judy Crystal. She died in February at age 76, leaving behind a loving husband of 50 years, 2 adult children, their spouses, and 6 grandchildren. Diagnosed just last November, she lived for a mere 4 months. She was also part of my extended family, mother of my first cousin's wife. She had a gift for making people feel loved and cared for. A speech and language pathologist, her own speech issues were the symptom that led to her diagnosis. She didn't even have time to get involved in the fight. Though her grandson, Micah, rode in the New York 5-borough bike ride with me on Sunday to raise money for Target ALS. And there are more. Jeff Grulich, who raised more than $1 million for Target ALS, died in April. As I was writing my remarks the other day, a close friend called to say that her brother had just passed away from ALS. And these are just the people I know and know of. It's enough. I start this way to remind you of the individuals who are out there, who are suffering with ALS, losing their ability to walk, to speak, to eat, and to breathe, who are dying. 5,000 to 6,000 people in the United States every year, 100,000 worldwide. Each and every one is precious. Each and every one leaves behind loved ones, work unfinished, potential unrealized. I start this way not just to remind you of the importance of your work, but of its urgency. I have my own personal reminder in Dan by my side every day. His presence and his continued focus on this fight remind me that we cannot stop until everyone with ALS lives. And going back to where I started, I am grateful for and so lifted up by this full room, 400 strong, with another 675 joining by video, and what you all represent. You represent the values of Target ALS, of radical collaboration. I have long believed in the power of people coming together to accomplish hard tasks, the seemingly unsolvable. Target ALS has adopted this value as one of its guiding lights. This room is a powerful representation of that value. You come from many different countries, bring diverse expertise to the mix. Our scientists in academia and industry, representatives of pharma, biotech, and venture capital, people with ALS, their caregivers, and the organizations that support them. Each of you is also precious for what he or she brings to this endeavor and for your commitment to working together. The value of deliberate disruption. We are not content with the status quo and are determined to break down the barriers that have stood in the way of progress. You all carry the DNA of that value into the world, coming together to look for new solutions in new ways, developing and using technology, crossing traditional boundaries. And the value of impatient optimism. We are not waiting hopefully for discoveries, but pushing ourselves and you to pursue the breakthroughs that will save lives. What you are going to present here over the next few days is the next installment of that drive that will fuel our optimism and catalyze more new ideas, discovery, experimentation, and hopefully move new drugs to clinical trials and to people. And this room represents real, deep, serious, exciting progress in understanding the basic biology of ALS, advancing knowledge of molecules and processes that drive the disease and that could stop it in developing biomarkers that can be used to diagnose and assess the efficacy of treatments, in gathering vital data from ALS patients worldwide, in analyzing the data that can lead to new insights and pathways for research, in the development of tools, technologies, and approaches that can unleash our ability to make new discoveries, find new and promising targets. I am here on behalf of all those who have ALS and who will be diagnosed with it. I'm here on behalf of all of those who love and care for them. I am here to celebrate your achievements, to soak in the hope that they give us. But I am also here to say directly and forcefully, please keep going, and please move faster. All of you, disruptors and collaborators, we are very, very impatient. Thank you, and have a great few days.
Good morning. I'm Amy Easton, VP of Scientific Programs for Target ALS. And today, I want to talk to you about removing barriers to precision medicine for ALS. I'm pretty sure that you'll leave the end with a better understanding of the unique innovation ecosystem that we're building, not only to break down traditional barriers in research, but to fundamentally transform the way that we do ALS research. So I think all of us here in this room can agree that one of the greatest challenges in developing effective therapeutics is our incomplete understanding of what causes the disease. With roughly 10% of people with ALS who have a known genetic mutation in a small number of genes, the remaining 90% do not have a clearly defined cause. And at the same time, we're beginning to appreciate that we have a heterogeneous population of individuals. Different subpopulations have different biological signatures and clinical manifestations of the disease. Inevitably, this can contribute to a higher rate of clinical trial failures and underscores the need for a more precise approach, one that enables us to develop targeted therapies tailored to the specific underlying biology of each patient. So to put precision medicine in practice, to deliver the right drug to the right patient at the right time in disease, we need to do the following things. We have to build tools. We have to build the diagnostic biomarkers to diagnose patients early. We need biomarkers to stratify these patients onto the right clinical trials. We need to build the rational therapeutic targets. We need drugs that have the right properties to allow for precise targeting of DNA, RNA, and protein. And finally, we need biomarkers that predict disease progression so we can intervene at the right time. So how as a community do we do this? I want to start by giving you an example of how Target ALS is approaching precision medicine with the most common genetic cause of ALS, the hexanucleotide repeat in the C9orf72 gene. So these are the tools that we're building. For diagnostic biomarkers, we're generating hi-fi long-read sequencing on every C9 case that we have. This allows for a more accurate measurement of the repeat length, methylation of the repeat, and phasing so that we can determine which allele contains the repeat. This helps facilitate development of new clinical diagnostics using long-read, and this helps facilitate the development of precision medicine. So for therapeutic targets, we're funding research. Many of you are here today to present your work on dissecting the toxic factors. In particular, at the end of 2025, we awarded a new consortium to bring forward groups together with complementary expertise to really tackle this. And Rosa Rademacher is leading that consortium, and they'll tell you more about that later this week. We're building preclinical models to also allow people to test your hypotheses. And many thanks to those of you who are helping to build those models, particularly to Regeneron for their generosity in contributing their C9 knock-in mouse model. We're funding, over the past many years, we've funded many different novel therapeutic modalities, from C9 ASOs to the sense strand to C9 antisense ASOs to CRISPR therapeutics. And all of you will hear more about that work today. Finally, we're generating an ever-growing longitudinal biomarker data set and biofluid data set from our natural history study to help all of you identify markers of disease progression. And together, all of this acts synergistically to advance precision medicine for C9. So now that I gave you an example of what we're doing for a specific known genetic mutation, how do we do this for the other 90% where there is no known cause? Target ALS is building an innovation ecosystem that powers the discovery of new therapeutics. And at the center of the engine is a collaborative funding model that brings together multidisciplinary teams, including academia and industry partners, to do translational research. Then we complement this by generating a detailed map of the disease through deep clinical and genetic phenotyping of people with ALS, carried out through AGRI, our ALS Global Research Initiative, our postmortem core, and strategic initiatives. These data are made broadly accessible through the data engine, a web-based federated platform that enables federated learning, and we're actively implementing new AI-powered tools to enable faster analysis and deeper interrogation of these high-dimensional data sets. And finally, through the Target ALS cores, we curate and provide access to hard-to-obtain resources that enable rigorous hypothesis testing. So historically, one iteration of discovery in the field can take years or maybe even a decade. But like a flywheel, we've already been doing the work and we already have momentum, and we can see that moving forward, we will be dramatically accelerating research and improve our success with every iteration of this cycle. So now, in the next 10 minutes, I want to share more with you about some of the details of this research infrastructure. So as I said, we fund multidisciplinary teams and top talent to fuel the drug discovery pipeline. So you can see on the slide here that currently we're funding 51 projects spanning 3 main areas of research: basic disease biology, drug discovery, and biomarker research. And with each new funding call, we are bringing in researchers new to ALS to continually foster new ideas and new collaborations. So you'll notice that our portfolio is weighted towards basic biology projects at the moment. That is intentional because we believe that understanding the fundamental mechanisms of the disease fuels the whole drug discovery pipeline. And we've recently just conducted a new round of funding to 6 new basic biology consortia, totaling another $2.8 million dedicated to this space. For the remainder of the year, we plan to expand our drug discovery and biomarker portfolio through our other funding calls that are ongoing. And so in the right-hand part of the slide, you can see the pie chart showing the different biological areas that we're investing in. And they range from TDP43 to cryptic exons, but they also include new areas of research, such as the peripheral immune system, lipid dysregulation, and pathways underlying risk and resilience. So in addition to the research we fund, we've been steadily expanding AGRI, our ALS Global Research Initiative, which is an integrated research framework that is comprised of 2 complementary studies: a natural history study. You can see the sites, the clinical sites indicated on the slide, as well as a community-based outreach study, with the goal of being the most comprehensive and inclusive study that there is to date. So these studies are designed to address fundamental questions in ALS biology, including the contributions of genetic and environmental risk factors and the variability in disease onset and progression across diverse populations. I want to highlight just a few of the exciting findings coming from this study. First of all, we have 426 participants enrolled across the 2 studies. The patient diversity continues to exceed historical studies of this kind, with about 35 participants coming from non-European descent. Second, in 2025, we generated our first batches of mass spec and O-lake proteomics data. We share those data out freely through the data engine, and this is already spurring biomarker development. You'll hear more about this in the poster session tonight and in tomorrow's session. And finally, I just want to mention that we've generated 180 hi-fi long reads to date, with the goal of reaching 6,000 over the next 5 years. And this taken together is really exciting because our preliminary look at the genetic data that we've generated shows us that we're detecting many new genetic variants in this cohort, particularly in the participants of non-European descent. So this is really starting to bear fruit. So switching gears to our postmortem tissue core, this is comprised of a group of clinicians and neuropathologists who work together to steward and curate tissue from postmortem from donors. And this postmortem tissue is, for each case, we're generating multimodal data. So this is an integrated research platform where we're sharing out the tissue alongside a rich resource of data sets. This year, with the help of our colleagues at Mount Sinai, we'll be able to share single-cell nuke-seq from 3 regions of the brain across 200 cases. With our help of our friends at Somagen and PacBio, we've generated more than 100 long reads to date, including on our 68 C9 cases. And with the help of CZI, we've generated the largest collection of uniformly stained phospho-TDP43 digitized images. And our friends at UCSF are already making progress developing a deep AI learning algorithm to quantify the staining and share these data and the algorithm openly with the research community. This is truly a unique resource that Target ALS has been building with all of you. So this is an alluvial plot just to highlight or underscore all of the big data sets that have been generated. These samples start by being collected at all of our sites on the left-hand side of the plot, our natural history study sites and our postmortem tissue core sites. They flow into our cores, and all of the data are generated with the help of our partners from Modality AI and Line of Speech, collecting digital health data, to New York Genome Center and Somagen generating the sequencing data. And again, all of these data are available for mining to all of you through our data engine, which makes all of these data easy to access and easy to mine. So this is a web-based platform that's hosted on DNASAC's Omics AI platform, making it possible to connect our data sets to Hi-Fi Solve's broader long-read data sets. So it enables AI-powered discoveries across distributed data sets. So the data engine today has 580 users worldwide, and similar to the way that email transforms how quickly and broadly we communicate with each other, the data engine has quickly amplified the impact of the data generated. So we're enabling researchers from around the world, you can see on the map, to mine the data, and the areas of investigation span basic disease mechanisms to biomarker discovery to therapeutic development. So in conclusion, I've just shown you that we're powering precision medicine for ALS with our innovation ecosystem by funding collaborative research and strategic initiatives, by enabling access to big data and key resources, and conducting clinical research on a global scale. And together, we're building a world where everyone lives. So thank you for your attention, and before I leave, I just want to acknowledge all of the people in this room who've been helping us build this ecosystem, from our PIs at our natural history study sites and our postmortem sites, to all of our partners generating the proteomics from the Zetterberg lab down to Somagen and DNA Stack, as well as all of the Target ALS board members, the staff, and our participants, the patients who are participating in our study. Thank you.